In 1854, a really terrifying and just deadly cholera outbreak swept through London. People were dying within hours of showing symptoms, and the medical establishment was, they were utterly paralyzed. I mean, they blamed miasma, right? Like bad air.
Right, which was the prevailing theory for almost everything back then.
Exactly. But one physician didn't look at the air, and he didn't even look at the patient's cells under a microscope. He just looked at a map. He carefully tracked the geographical distribution of every single cholera case. He interviewed the healthy and the sick and traced the entire outbreak back to one infected public water pump on Broadwick Street.
Yeah, John Snow, it's really one of the most powerful stories in medical history because it proves something profound,
He just took the handle off the pump and the outbreak stopped.
Right. It proves that you can track, predict, and prevent a disease across a massive population even before you understand its biological mechanism. Because, I mean, at that time, nobody knew what a bacterium was, yet by observing those population patterns, the threat was neutralized.
Welcome to our deep dive. If you're listening right now, we know you are the learner. Whether you're like a medical student prepping for boards, a clinician trying to connect the dots, or just a fiercely curious, self-taught intellect. Today, we are immersing ourselves in the textbook Pathology and Epidemiology of Cancer.
Which is an incredibly dense but rewarding text.
Oh, absolutely. And our mission today is to explore how we bridge the gap between that macro-level population map, so epidemiology, and the micro-level behavior of a single rogue cell, which is pathology. We're going to see how these two distinct disciplines are merging to basically decode the mechanics of cancer.
Which is necessary, right? Because cancer is not, it's not a straightforward binary diagnostic event. When you're looking at a fractured bone on an X-ray, the pathology is obvious. The bone is broken. But tumor biology is notoriously complex.
Yeah, you can't just slap a cast on it. Before we even look at a cancer cell, we need to understand how we track diseases across massive populations.
Right, and to unravel it, epidemiologists rely on three foundational metrics to map the burden of disease. We've got incidence, prevalence, and mortality.
I always like to visualize these metrics because keeping them distinct is just, it's crucial for reading the literature. I always picture a bathtub.
A bathtub, okay, I like where this is going.
Yeah, so incidence is the water flowing out of the faucet and into the tub. It represents the rate of new cases occurring within a specific time period. So when our source text notes that, say, the global incidence of stomach cancer for men in 2012 was about 18 per 100,000 per year, that's our flow rate from the faucet.
Right, that makes sense.
And prevalence then is the total amount of water sitting in the tub at any given moment. So the number of people actively living with the disease.
The bathtub visual works really well for the basic math, but clinical biology is always a bit messier.
Of course it is.
Think of that bathtub as actively leaking. That leak is mortality, the rate of deaths. But here's the kicker. It's not just deaths from the cancer itself. It's deaths from all causes.
Oh, wait, so if someone dies of something totally unrelated, it still drains the tub.
Exactly. This is a vital concept in epidemiology called competing risks. Say you have a patient with a very slow-growing prostate tumor, but they die of a sudden myocardial infarction, a heart attack. That water still leaves the tub. It lowers the prevalence of the cancer in the population, even though the cancer wasn't the cause of death.
That makes a lot of sense. It's a dynamic system. And to measure these shifting rates, epidemiologists use specific study designs, right? Like we have cohort studies, which follow a specific group of people forward in time to see who develops a disease.
Yes, forward-looking.
Then we have case control studies, which work backward. You take people who are already sick, the cases, and compare their past exposures to healthy people, which are the controls.
Which, by the way, is the absolute gold standard for studying rare outcomes.
Oh, really? Why is that?
Well, think about it. If a specific sarcoma only occurs in, like, 1 in a million people, a cohort study would require following tens of millions of people for decades just to find a handful of cases. A case control study completely bypasses that logistical nightmare because you start with the sick patients.
Right, you already have the cases. But then there's the third type, the ecological study. These look at data on a much broader population level, usually based on, geographical regions or time periods, rather than individual patient data.
Yeah, the macro view.
And this is where I kind of get skeptical. If an ecological study lacks individual data, how does it avoid just pointing out pure coincidences? Like, I don't know, people in France consume a lot of cheese, and they also have higher rates of lung cancer, so obviously cheese causes lung cancer.
You're hitting on the exact limitation of the design. It's literally known as the ecological fallacy.
Ah, so there's a name for it.
Yes, ecological studies absolutely cannot prove biological causation. They are completely vulnerable to confounding variables. Like your example, the fact that populations eating more cheese might also just have historically higher rates of smoking.
Right. The cheese has nothing to do with it.
Exactly.
Yeah.
The role of an ecological study isn't to prove a cause, it's to generate a hypothesis. It flags a statistical association. But the fundamental rule of epidemiology is that statistical association does not equal biological causation.
So the epidemiologists are essentially like detectives drawing a perimeter around a neighborhood and saying, there's a high rate of crime happening here.
That's a great way to put it.
But to actually prove how the crime is happening and who is doing it, we have to transition from the population map to the tissue itself. We have to look under the microscope.
Precisely. We need pathology, which comes from the Greek pathos, meaning suffering, and logia, meaning study. In cancer, pathologists are primarily analyzing neoplasia, the formation of new, abnormal tissue growth.
Okay. And the critical clinical divide here is determining whether that neoplasm is benign or malignant. We all know benign means it hasn't spread, and malignant means it's aggressive.
Right. That's the common understanding.
But I want to really dig into the actual physical mechanism. What is a malignant cell physically doing that a benign cell isn't?
It comes down to structural boundaries, specifically the basement membrane.
The basement membrane.
Yeah, it's a dense, sheet-like layer of extracellular matrix that basically anchors epithelial tissues to the underlying connective tissue.
Okay, so it's a foundation.
Exactly. Now, a benign tumor grows, it expands, it might even compress nearby organs and cause severe symptoms, but it respects that boundary. It stays encapsulated. Think of a lipoma, a benign tumor of fat cells. It just sits there.
It's kind of like a bad neighbor who builds an ugly shed, but they stay on their own property.
Yes. But a malignant tumor, however, actively secretes enzymes like matrix metalloproteinases that literally digest and degrade that basement membrane.
So it's not just growing, it's actually equipped with chemical weapons to bore through the tissue's physical defense lines. It's the neighbor who breaks down your fence and starts living in your living room.
Unfortunately, yes.
Yeah.
And once it breaches that membrane, it gains access to the blood vessels and the lymphatic system. That provides the highway for metastasis spreading to distant organs.
Right, the spread.
And how these malignant tumors behave largely depends on their embryonic origins. Our text categorizes them heavily by their germ layer. Carcinomas, which make up the vast majority of human cancers, arise from epithelial cells. So the tissues lining our organs and skin.
And then we have sarcomas, right?
Right. Sarcomas arise from mesenchymal cells, the structural tissues like bone, muscle, and fat.
Why does that origin matter, though? Like, for the patient's prognosis or treatment, does a sarcoma spread differently than a carcinoma?
It often does, yeah, because sarcomas originate in deep connective tissues that are rich in blood supply. They frequently metastasize through the blood, which is called hematogenous spread, and they often land in the lungs. Whereas carcinomas, originating in epithelial linings, tend to spread first through the lymphatic system, which is exactly why checking the local lymph nodes is so critical for a breast or colon carcinoma.
That makes total sense. And then we have lymphomas and leukemias, which arise from the hematopoietic or blood-forming cells. So a pathologist looks at the slide, identifies the tissue origin, and confirms it has breached the basement membrane.
Correct.
But tumors aren't all equally aggressive. I want to talk about tumor grade. How do pathologists actually quantify the aggression of these cells under the lens?
They look at cellular differentiation, meaning how closely does this mutated cell still resemble the normal, healthy cell it originated from? A low-grade tumor is well differentiated.
So it looks pretty normal.
Right. The cells are mutated, yes, but they still form recognizable glandular structures or tissue patterns. But a high-grade tumor is poorly differentiated.
Meaning the cellular architecture has just completely devolved into chaos.
Complete chaos. Pathologists look for very specific visual red flags. First is pleomorphism. The cells and their nuclei vary wildly in size and shape.
They're all mismatched.
Yeah. Next, they look for hyperchromatic nuclei. The nuclei stain incredibly dark on the slide because the cell is hoarding massive amounts of duplicated DNA, frantically preparing to divide.
And they actually count the divisions too, don't they?
They do. They count mitotic figures, literally catching the cells in the act of division. The more mitotic figures, the faster the tumor is growing.
And if it grows too fast, it creates its own physical limitations, right? Like the text mentions necrosis.
Yes. In a highly aggressive high-grade tumor, The cells divide so rapidly that the tumor mass literally outgrows its own blood supply. The cells in the center of the tumor starve to death and die, leaving a necrotic core.
Wow.
When a pathologist sees extensive necrosis on a slide, it's a glaring indicator of a highly aggressive, rapidly proliferating malignancy.
So we have the epidemiologists finding the statistical associations in the population and the pathologists identifying the aggressive architecture in the tissue.
Right, the macro and the micro.
Which brings us to a massive turning point in the textbook. The synergy of these two fields, a discipline known as pathoepidemiology.
Yeah, and the 1983 passive smoking study is the textbook's classic example of this synergy.
Oh, I found this part fascinating.
Right. So epidemiologists had already flagged an association. Non-smoking women married to men who smoked had a significantly higher risk of lung cancer. But critics argued it was just an association. Maybe these women shared other environmental factors, or maybe, their diets were different.
Because association isn't causation. To prove causation, you need the tissue.
Exactly. So pathologists stepped in. They performed blind autopsies on 283 adults who had died of completely non-cancer-related causes, like car accidents or heart disease.
And they didn't know who lived with a smoker and who didn't.
Nope. The pathologists examined the lung tissue blind, and what they found were pre-neoplastic epithelial lesions, these early microscopic architectural damages. In the passive smokers, that perfectly mirrored the damage found in active smokers.
That is incredible. They caught the biological mechanism in its infancy before it even became a full-blown malignancy. They completely bridged the gap between the population survey and the cellular reality.
They did. And this synergy doesn't just prove causes, It completely redefines the diseases we thought we knew. Let's look at breast cancer.
Yeah, gene expression profiling has proven that calling it just breast cancer is a terrible, overly broad term. It's not one disease.
Not at all. It is a collection of genetically distinct subtypes. Some are driven by estrogen receptors, some by HER2 protein amplification. Once pathologists firmly established these molecular subtypes, epidemiologists had to, completely reevaluate their population data.
Because the old data was treating all breast cancers as the same thing.
Right. A great example from the text is the study of dietary carotenoids, like lycopene, which is found in tomatoes.
I'm guessing the initial population data probably showed a modest or confusing protective effect against breast cancer as a whole.
You're exactly right. If you study an exposure across all breast cancer patients, the protective effect just gets diluted. But when epidemiologists stratified the data by the new pathological subtypes, a clear signal emerged.
What did they find?
Higher levels of circulating carotenoids were associated with an 18 to 28% lower risk, specifically for estrogen receptor negative breast cancers.
Wow.
Yeah, and those ER-negative cancers are typically much harder to treat.
That is a staggering shift in perspective. It means treating colon cancer or breast cancer as a single biological entity isn't just outdated, it's scientifically inaccurate.
Completely inaccurate.
And speaking of colon cancer, the textbook breaks down colorectal cancer using the Fearon and Vogelstein model from 1990. They mapped out the step-by-step genetic mutations that transform a healthy colon cell into a benign polyp and eventually into an invasive carcinoma.
That model was groundbreaking. It laid the foundation for our current understanding that colorectal cancer develops through 3 primary molecular pathways.
What's the first one?
The most common, accounting for about 80% of sporadic cases, is the chromosomal instability pathway, or CIN.
Okay, CIN. How does that work?
It's essentially a massive failure in mitosis. Whole sections of chromosomes are duplicated or lost during cell division, leading to massive genetic imbalances, which we call aneuploidy.
So the cell's basic instruction manual is basically missing entire chapters or has chapters duplicated 10 times over. What are the other 2 pathways?
The second is microsatellite instability, or MSI. Microsatellites are short, repeating sequences of DNA. In healthy cells, if a mistake is made during DNA replication, we have mismatch repair enzymes that act like a, genetic spell checker.
Okay, so they fix the typos.
Right. But in MSI, that spell checker is broken. Mutations rapidly accumulate in these repeating sequences.
And the third pathway, this one sounds heavily driven by epigenetics, right?
Yes, exactly. The CpG island methylator pathway, or CMP. This accounts for about 10 to 15% of cases.
CMP, got it.
This isn't about the DNA sequence mutating or breaking. It's about methylation, the addition of chemical tags to the DNA. Imagine pouring superglue over a light switch.
Oh, wow. Okay.
In CMP, hypermethylation acts like that superglue. It effectively padlocks the off switch on crucial tumor suppressor genes.
Yeah.
The underlying DNA sequence is perfectly normal, but the gene is chemically silenced.
So the gene is there, but the cell just can't read it.
Exactly.
Understanding those specific pathways, CIN, MSI, CMP, is crucial because they dictate how the tumor responds to chemotherapy or immunotherapy, right?
Absolutely.
But we
Yeah.
can't just talk about the mutated tumor cells in isolation. Cancer cells don't build a tumor on their own. They have to manipulate the entire neighborhood around them to survive.
This brings us to the tumor microenvironment, or the
Okay.
stroma. Normal tissue is a highly structured framework of connective tissue, blood vessels, and immune cells. A malignant tumor doesn't just, push this tissue aside. It recruits the local structural cells, called fibroblasts, and biochemically brainwashes them
Brainwashes them into what?
into becoming cancer-associated fibroblasts, or CFs.
It's essentially like an invading army that arrives with no supplies. Instead of bringing its own logistics, it captures the local townspeople and forces them to build fortresses and farm food for the enemy troops.
That is an excellent way to conceptualize the desmoplastic reaction.
The desmoplastic reaction.
Yeah, it's the dense, fibrous scar tissue that these hijacked fibroblasts build around tumors, particularly in breast and pancreatic cancers. This fibrous wall makes it incredibly difficult for chemotherapy drugs to even physically reach the tumor cells.
And the townspeople aren't just building walls, right? They're building roads. Let's talk about angiogenesis.
The tumor cannot grow beyond a few millimeters without a dedicated blood supply to deliver oxygen and nutrients, so it secretes chemical orders. Most notably, VEGF, vascular endothelial growth factor.
VEGF.
Yes, this molecule acts as a signal to the surrounding tissue, instructing it to sprout new, chaotic networks of blood vessels directly into the tumor mass.
But because we understand this pathology now, we can create drugs that target the townspeople, right? We don't just poison the tumor, we cut off the logistics.
Exactly. This is the basis of targeted therapies. Drugs like bevacizumab are monoclonal antibodies designed to specifically bind to and neutralize that VEGF molecule.
So it intercepts the signal.
Right. By intercepting the chemical orders, the new blood vessels never form, and you literally choke off the tumor's blood supply. Other experimental therapies target the cancer-associated fibroblasts directly to break down that fibrous desmoplastic wall we just talked about.
Okay, so the tumor secures a blood supply, but how does it process the nutrients it receives? This leads us to a really fascinating metabolic shift discovered by Otto Warburg in the 1920s, the Warburg effect.
Yeah. Normal, healthy cells are incredibly efficient at making energy. In the presence of oxygen, they completely burn glucose through oxidative phosphorylation, generating a massive amount of ATP, which is the cell's energy currency.
Makes sense.
But cancer cells, even when they have plenty of oxygen, switch to a process called aerobic glycolysis.
Wait, I remember from biochemistry that glycolysis is incredibly inefficient compared to oxidative phosphorylation. It produces a tiny fraction of the ATP.
It does.
Why would a rapidly dividing, energy-hungry tumor cell choose a less efficient method of making energy? That seems backward.
Yeah, it does seem backward until you realize a cancer cell's primary goal isn't just generating energy, it's generating biomass.
Biomass.
Yeah. It needs to physically build two daughter cells. If it completely burns all the glucose down to carbon dioxide for maximum energy, there's no carbon left over to build the physical structures of the new cells. Aerobic glycolysis only partially breaks down the glucose. It leaves an abundance of intermediate carbon chains. The cancer cell uses those chains as the raw building blocks to synthesize lipids, proteins, and nucleotides. It sacrifices energy efficiency for raw construction materials.
That is deviously brilliant. It's fundamentally rewiring the cell's metabolism. But here's the catch, right? Not every cell in that tumor is undergoing the exact same rewiring or sharing the exact same mutations.
No, they're not.
This introduces the concept of intratumoral heterogeneity. A single tumor is a mosaic of different cell populations. The text contrasts two major theories for how this happens. Let's start with the first one.
The first is the clonal evolution theory. This is essentially Darwinian evolution on a microscopic scale.
Okay, survival of the fittest.
Right. As cells rapidly divide, they make random genetic mistakes. Most mistakes are lethal to the cell, but occasionally a cell acquires a mutation that gives it a survival advantage. Maybe it develops resistance to hypoxia-low oxygen. That cell survives, replicates, and creates a dominant subclone within the tumor.
So in this model, virtually any cell within the tumor has the potential to accumulate the right mutations and drive new growth.
Yes, any cell could potentially become the driver.
But if any cell can drive the tumor, why do we see patients undergo, really intense chemotherapy that wipes out 99% of the tumor mass, only for the cancer to return aggressively months later? Are some cells fundamentally more resilient from the very beginning?
And that question is the foundation of the competing theory, the cancer stem cell theory.
How does that one work?
This model proposes a strict hierarchy, kind of like a beehive. It suggests that the vast bulk of the tumor cells are like worker bees. They divide rapidly, they make up the physical mass, but they have a limited lifespan, and they actually cannot initiate a completely new tumor.
So who's initiating it?
Only a tiny, specific subset of cells, the cancer stem cells, the queen bees, so to speak, possess the unique ability to self-renew indefinitely and seed new tumors.
Which completely changes the therapeutic target. If you use a drug that rapidly kills all the fast-dividing worker bees, the tumor shrinks on the MRI, and it looks like a clinical victory.
But if that drug doesn't kill the dormant, slow-dividing queen bee...
The tumor will inevitably rebuild itself.
Exactly. Understanding which of these theories, or which combination of them, is operating in a patient-specific cancer dictates whether we need therapies targeting rapid vision or therapies targeting stem cell pathways.
So we're dealing with tumors that manipulate their environment, rewire their metabolism, and evolve dynamically. It brings up a crucial question about the origins of all this chaos. How much of this biological blueprint is hardwired into our DNA before a tumor even forms?
That's where genetic epidemiology comes in.
Let's look at that. To untangle what is inherited in our DNA versus what is caused by our environment, epidemiologists rely on twin studies, right?
Right. By comparing disease concordance rates in monozygotic twins, so identical twins who share 100% of their DNA against dizygotic or fraternal twins who share roughly 50% of their segregating genes.
So you can mathematically isolate the genetic component of a disease.
Yes.
The formula the text uses is pretty straightforward, but really powerful. The genetic contribution equals 2 times the difference between the correlation in identical twins and the correlation in fraternal twins.
Exactly. You calculate the difference in correlation between the two groups, which represents that 50% gap in shared genetics, and then you just multiply by 2 to extrapolate the full 100% genetic effect.
Got it. And when this math was applied to massive populations, like the Nordic twin study of cancer, the results really quantified our risk. They found that breast cancer is about 27% heritable. Colorectal cancer is 35% heritable.
But the data on prostate cancer underwent a really fascinating revision.
Right. Initially, the heritability of prostate cancer was calculated at 45%, but recent estimates pushed that heritability all the way up to 58%. How did the genetics of the disease suddenly change?
Well, the genetics didn't change. Our epidemiology improved. Remember our bathtub analogy earlier? And the concept of competing risks.
Oh, the leaky tub where people die of a heart attack instead.
Yes. Prostate cancer often develops late in life. In older studies, many men who were genetically susceptible to prostate cancer died of heart attacks or strokes in their 60s before their prostate cancer was ever clinically diagnosed.
Oh, I see. Because their cancer was never recorded, the identical twin correlation looked artificially low.
Precisely. The math was fundamentally flawed because the patients didn't live long enough to express their genetic destiny.
Wow.
When modern epidemiologists applied time-to-event analyses, these are statistical models that actually account for those competing causes of death. They revealed the true genetic influence. Prostate cancer is highly heritable, up to 58%.
That's huge. But even with a 58% heritability, that still leaves 42% of the risk up to random chance and the environment.
Exactly.
Inheriting a susceptibility gene doesn't mean you are guaranteed to get the cancer. It just means your biological threshold for developing it is significantly lower. Every environmental exposure you accumulate over your lifetime still interacts with those genes.
Which is why pathoepidemiology is so critical. Nature sets the trap, but the environment springs it.
That's a great way to say it. And when it finally is sprung and a patient is sitting in a clinic waiting for their diagnosis, all of this complex biology, the germ layers, the subtypes, the genetic pathways, it all has to be distilled into an actionable treatment plan.
And that is the role of the TNM staging system.
TNM stands for tumor, nodes, and metastasis. The T evaluates the physical size of the primary tumor and how deeply it has invaded the local tissue, right? Like whether it's breached that basement membrane and pushed into nearby structures.
Correct. And the N assesses the regional lymph nodes. If cancer cells have reached the nodes, it indicates the tumor has gained access to the lymphatic highway.
And finally, the M determines if there is distant metastasis, if those cells have set up colonies in the liver, lungs, or the brain.
But there is a really crucial clinical distinction to make here between the clinical stage, which is marked with a lowercase c on a patient's chart, and the pathologic stage, which is marked with a lowercase p.
Okay, what's the difference?
Well, Clinical staging is our best educated assessment based on pre-surgical data. So physical exams, MRI scans, needle biopsies, it guides the initial treatment plan.
But a scan can't show microscopic invasion, can it?
It cannot.
Yeah.
That's exactly why pathologic staging PT&M is the gold standard. It is determined only after the surgeon has excised the tumor and the surrounding lymph nodes, and the pathologist has actually evaluated the tissue under the microscope.
Pathologic staging doesn't rely on imaging shadows. It relies on holding the biological truth right there in your hands.
Exactly right.
Which really brings us full circle today. We've traveled from the macro flow of populations on Broderick Street with Jon Snow to the microscopic reality of a hyperchromatic nucleus and a degraded basement membrane.
It's quite a journey.
It is. We've seen how tracking the population map points us toward the biological reality, and how redefining the biological reality forces us to redraw the population map.
Because beating cancer demands both lenses. You have to zoom all the way out to understand the exposures and zoom all the way in to decode the mechanisms.
And as promised, for the medical learners listening to us today, let's do a quick mental review to lock in what we've covered. Based on today's deep dive, I want you to imagine a patient is diagnosed with a high-grade sarcoma. Take a moment on your own to break down exactly what high-grade means regarding cellular differentiation and mitotic figures.
That's a good one.
Then define what sarcoma implies about the tumor's germ layer origin and its likely pathway of spread. Finally, consider how you might design a retrospective case control study to identify the environmental risk factors that may have interacted with that patient's genetics.
It's the ultimate test of integrating pathology and epidemiology. If you can answer that, you really grasp the material.
You definitely did. We started today by talking about how cancer diagnosis used to be this incredibly murky, muddy landscape compared to a simple broken bone. But we aren't just staring at the mud anymore. We're mapping it down to the specific molecular pathways and the methylated DNA.
The progress is astounding.
Which leaves me with a final provocative thought for you to chew on. If molecular pathology is advancing to the point where we classify tumors strictly by their genetic signatures, like the CIN or CMP pathways we explored today, will traditional organ-based classifications eventually become obsolete?
Oh, that's a profound question.
Right. In 20 years, will we stop telling patients they have lung cancer or breast cancer and instead diagnose and treat them entirely based on the mutated pathway, regardless of where the tumor happens to be physically located? Something to think about. Until next time, keep learning.